All right, let's dive deep today. We're going to be tackling bioanalytical method development.
Oh, exciting.
Now, before you think boring, I promise you this is about way more than just like test tubes and beakers.
Right.
This is about how scientists figure out how to measure drug levels in your body, things like blood and urine, with crazy accuracy.
Yeah.
And why should you care? Well, this is how we know a drug is working, how much to give you, and if it's safe.
Exactly. What gets me really excited about this is think about the challenge of measuring these tiny, tiny amounts of drug often hidden within a complex biological soup.
Oh, okay.
It's like finding a specific grain of rice in a giant paella. That's why we need these really sensitive and reliable methods.
Okay, I'm getting hungry, but I get your point.
Yeah,
So it's not just about detecting the drug, but it's about measuring it precisely.
absolutely. You need to know exactly how much drug is present in the sample. And to do this, we develop assays, highly specialized tests designed to pick out our target drug and measure it with crazy accuracy.
So it's like a recipe specifically designed to find and quantify that one ingredient. But how do we know these assays are even reliable? We're talking about making decisions about your health based on these measurements.
Right, so that's what validation comes in. It's a rigorous process where we put these assays through a ton of tests, like a battery of tests, to prove that they're up to the task. Think of it as a quality control boot camp for assays.
Boot camp, huh? Intense. So what kind of drills are we talking about here?
Well, we need to make sure that the assay is accurate, that it's hitting the bullseye, meaning it measures the true drug concentration. Imagine you step on a scale and it shows you weigh 20 pounds less than you actually do. Not very helpful, right?
Not only not a confidence booster.
Yeah.
So accuracy is crucial. What else?
Precision is just as important. This means the assay consistently hits the same spot on the target every time. Let's say you're baking a cake and your recipe, the assay, calls for a precise amount of flour. If you're a little off each time, you'll end up with some pretty inconsistent cakes.
Okay, so accuracy is about getting it right, and precision is about getting it the same every time. Consistency is key. What other tests are there?
We also test for sensitivity and specificity.
Okay.
Sensitivity is the ability to detect even the smallest trace of the drug, kind of like a bloodhound sniffing out a single scent in a crowded room.
Wow, so even tiny amounts won't escape these tests. That's impressive. What about specificity?
Specificity is about making sure the assay only picks up our target drug and doesn't get confused by other substances in the sample. Imagine trying to find a specific book in a library with millions of books. If your search isn't specific enough, you'll get a ton of irrelevant results.
Makes sense. So we need an assay that's like a super sleuth, finding our target drug accurately, precisely, sensitively, and specifically. That's a lot to ask.
It is, but the stakes are high, especially when we talk about something like drug development.
Speaking of which, this whole process must be pretty tightly regulated, right?
Yes.
I mean, we're talking about medications that people are going to be using to treat people.
You're absolutely right. Organizations like the FDA and the ICH set strict standards for bioanalytical method development and validation. They're like the referees making sure everyone's playing by the rules, which is incredibly important when we're talking about patient safety.
So it's not just about the science. It's about ensuring these methods are reliable and meet the highest standards.
Yeah.
But I'm curious, how do these validated methods actually get applied in real-world drug development scenarios?
That's a great question, and that's exactly what we'll delve into next. We'll explore how these validated methods help scientists figure out the best way to give you a drug, whether it's a pill, an injection, or something else, and how they play a crucial role in ensuring that new medications are safe and effective. Welcome back, deep divers.
You know, it's kind of mind-blowing to think that such tiny measurements can have such a huge impact on drug development. So what are some real-world scenarios where these methods are just absolutely critical?
Well, one crucial area is figuring out the best way to give you a drug. Is it a pill you swallow, a cream you rub on your skin, an injection? Bioanalytical methods help us determine which delivery method will get the drug to where it needs to be in your body and at the right concentration.
So it's like finding the perfect vehicle to transport the drug to its destination in your body.
Yeah.
But there are a lot of factors to consider, right? Not all drugs behave the same way.
Exactly. Some drugs are easily absorbed into the bloodstream, while others might need a little help. And this is where understanding a drug's pharmacokinetic profile comes in.
Okay, remind me, what exactly is a pharmacokinetic profile? Sounds a little intimidating.
Oh, it's actually simpler than it sounds. Pharmacokinetics is basically the study of what your body does to a drug, how it gets absorbed, distributed throughout your body, metabolized, and eventually eliminated. Think of it like tracking a package's journey from the warehouse to your doorstep.
I like that analogy.
Yeah.
So, by measuring drug levels in your blood or other samples, scientists can map out this journey and see how the drug is behaving in your body.
Precisely. And this information is incredibly valuable when it comes to determining the optimal dosage form. For example, let's say we have a new pain reliever that's showing great promise. We need to figure out the best way to deliver it as a pill, a liquid, or maybe even a patch.
So, scientists would give the drug to volunteers in different forms and then use these bioanalytical methods to track how much drug ends up in their bloodstream over time.
Exactly. And by comparing the results, we can see which dosage form delivers the drug most effectively and consistently. We're looking for that Goldilocks zone, not too much, not too little, but just the right amount of drug to be effective without causing unwanted side effects.
That makes sense. So, bioanalytical methods are like the detectives helping us solve the mystery of how a drug moves through the body.
That's a great way to put it. And it's not just about efficacy, getting the drug to work, it's also about safety.
Of course, safety is paramount. How do these methods help ensure that new medications are safe for people to take?
Well, before a new drug can be approved for use, it undergoes rigorous safety testing, often involving animal studies. By analyzing blood and tissue samples, we can detect even subtle changes that might indicate a problem. It's like having a super sensitive early warning system that can alert us to potential issues before they become serious.
So bioanalytical methods are like the guardians, looking out for any signs of trouble and making sure that new drug is safe enough to move forward in development.
Precisely. And this kind of monitoring continues even after a drug is approved and available to the public.
Oh, really? I thought once a drug was approved, that was it. Why is continued monitoring so important?
Because people are diverse, and not everyone responds to a drug in the same way. Factors like age, genetics, other medications you might be taking, even your diet can influence how your body processes a drug.
So it's not a one-size-fits-all situation.
Yeah.
That makes a lot of sense. But how do we account for all these variations?
That's where the concept of personalized medicine comes in. It's the idea that treatments should be tailored to each individual, taking into account their unique characteristics.
That sounds amazing, but also incredibly complex. How can we possibly personalize treatments for every single person?
It's a challenge, but one that's becoming increasingly achievable thanks to advances in fields like pharmacogenomics and the study of the microbiome.
Pharmaco what? You're going to have to break those down for me. They sound like something out of a science fiction movie.
Okay, let's unpack those terms. Pharmacogenomics is the study of how your genes affect your response to drugs.
So our genes can actually influence how well a drug works for us. I had no idea.
Absolutely. We're learning that even small variations in your DNA can make a big difference in how your body metabolizes a drug. This means that some people might need a higher or lower dose of a medication to get the same effect, while others might be more prone to side effects.
Wow, that's fascinating. And what about the microbiome? I know it has something to do with the bacteria in our gut, but how does that tie into drug response?
It turns out that the trillions of bacteria living in our gut play a surprisingly important role in our health, and that includes how we respond to medications. Some of these microbes can actually metabolize drugs, altering their effectiveness.
So it's like having a whole ecosystem of tiny pharmacists living inside us, helping or hindering the way drugs work.
It's a great analogy, and bioanalytical methods are playing a crucial role in helping us understand these complex interactions. By studying the microbiome and how it influences drug metabolism, we can develop more targeted and effective therapies.
It's amazing to think that something as seemingly simple as measuring drug levels can lead to such groundbreaking insights. It really underscores the power of scientific curiosity and the importance of constantly pushing the boundaries of knowledge.
I couldn't agree more. And as we delve deeper into these emerging fields, bioanalytical methods will continue to be essential tools in shaping the future of medicine. But for now, let's take a moment to recap what we've learned about these fascinating techniques and the crucial role they play in drug development.
All right, so we've journeyed through this intricate world of bioanalytical method development. We've seen how these incredibly sensitive and precise measurements are like the unsung heroes of drug development, ensuring both safety and effectiveness.
Right.
But before we wrap up, I have to ask, where do we go from here? What does the future hold for this field?
Oh, the future of bioanalytical method development is incredibly exciting. As we move towards personalized medicine, these methods will become even more critical. Imagine a world where your doctor can prescribe the exact right dose of a medication based on your individual genetic makeup and your gut microbiome profile.
That sounds like science fiction becoming reality. but I can see how bioanalytical methods would be essential for making that vision a reality.
Absolutely. These techniques are the key to unlocking a deeper understanding of how drugs interact with our bodies on a personalized level. We're no longer just talking about measuring drug levels. We're talking about using those measurements to predict individual responses and tailor treatments for maximum effectiveness and minimal risk.
It's like having a personalized roadmap for each patient's journey with a medication. So cool. But I imagine there are still challenges ahead. What are some of the hurdles that scientists are facing in this field?
One of the biggest challenges is developing even more sensitive and specific assays.
Hmm.
We need to be able to detect and quantify drugs with incredible precision, even when they're present in tiny amounts or in complex biological mixtures.
So it's like trying to find a needle in a haystack, but the needle is microscopic and the haystack is constantly shifting.
That's a great analogy. But advancements in technologies like mass spectrometry are really helping us overcome these obstacles. These incredibly powerful tools allow us to analyze complex samples and identify individual molecules with incredible accuracy.
Wow, it sounds like we're entering a whole new era of precision in medicine.
We are, and as these technologies continue to evolve, we'll be able to unlock even more insights into the complex interplay of drugs, genes, and the environment.
It's exciting to think about the possibilities, but as with any powerful technology, I imagine there are ethical considerations as well.
Absolutely. As we gain a deeper understanding of individual drug responses, it's crucial that we use this knowledge responsibly and ethically. We need to ensure that access to personalized medicine is equitable and that patient privacy is protected.
That's a good point. It's a reminder that even the most groundbreaking scientific advancements need to be guided by ethical principles.
Well said. It's a responsibility we all share.
This has been an incredible deep dive into a field that's often hidden from view, but is so fundamental to our health and well-being. We've explored the intricate science behind measuring drug levels, the rigorous processes involved in validating these methods, and the profound impact these techniques are having on drug development and patient care.
It's been a pleasure sharing these insights with you. And to our listeners, we hope you've gained a newfound appreciation for the crucial role that bioanalytical method development plays in shaping the future of medicine.
So next time you take a medication, remember the incredible science and the dedicated scientists working behind the scenes to ensure that it's safe, effective, and tailored to your individual needs.
Until next time, keep exploring, keep learning, and keep diving deep.